Greetings everyone. Let me see that my camera is everything is on. Yes. Okay. Greetings, one and all, and welcome to another episode on the channel. Although today we have some sad news. This week we start with a sad remembrance actually. We lost Professor Roman Carlos, an oral pathologist from Guatemala, who, while I did not know him personally, besides the typical greetings of conferences, he always responded with, I must say, with much cheer and very open and charming self that he was. But we have lost him, and from whatever I have heard from everybody else, there has been a lot of news about him, a lot of thought, a lot of feelings that have been shared. And from what I have heard, is the world not just lost a very great oral pathologist, but also a very great and kind human being. We will remember him. He will, like many other greats of our field, will always be there in our thoughts. And well, we just, as is expected, we do have to go on and I guess honor their memory by just going on doing the best that we can. And on that note, today we have a session on saliva, which in so many ways is the unrecognized and unappreciated keeper of the oral cavity. Saliva is really our thing as dentists, and yet somehow we don't really pay it much attention, sadly. It sort of takes, it does not take the place of pride that it should, but well, we're trying to solve that situation, at least in a very small step today. So, we are going to discuss everything about diagnostics and therapy where saliva comes in. For that, we have with us Dr. Krishnaraj, who has been actually working on this topic for quite some time, and he's going to share not just theory but his actual practical experiences, which you can put in place almost immediately in your practice too. So, Dr. Krishnaraj Ganesh Narayan, Professor, Department of Periodontics, and Head Office of Safety and Infection Control, Core Member, Institutional Review Board, Kurg Institute of Dental Sciences, Virajpet. He is a healthcare professional, a surgeon, an entrepreneur, a researcher, a mentor, an academician with over 17 years of experience in providing clinical periodontal care and administering patient care services. Quality assurance is really his thing, and he has also done a lot of translational research. He's a founder of the Delta Dental Care and Dental Care Academy and founding partner of Dental Care. Okay. Sorry. We were privy to his excellent knowledge on infection control sometime back when he was on the channel as the moderator for this session on COVID-19 vaccine and the dental team. Welcome back, Dr. Krishnaraj. It's a pleasure to have you here.
Thank you. Thank you,
And
ma'am.
you're welcome. We are also joined by Dr. Maribasappa Karchet. He is the expert for today's session. Dr. Karchet is Associate Professor of Oral Microbiology and Faculty of Dentistry, Kuwait University. He has his PhD from Sweden and a postdoctoral fellow at Rutgers School of Dental Medicine, US. He is on the editorial board of Frontiers in Cellular Infection Microbiology, Frontiers in Oral Health, British Medical Journal, and Microbiology Ambassador for the American Society of Microbiology. Welcome, Dr. Maribasappa, and welcome again to both of you and to all our
Thank
viewers.
you.
Yes, you're most welcome. So now, today, we have a little change in our regular. Let me first stop sharing this. One second. So, our regular plan is the presentation is live. But today, in fact, we don't have a live presentation. It is recorded, although Dr. Krishnaraj is with us and he will be, of course, interacting at the end. But due to expected problems with network, we have his presentation as a recording, which now I will just add to the stream. And one second. So we can all watch that. And why is that not working? Okay, sorry. I need to take a second and get this organized. I guess I need to stop this other share.
At the outset, I would like to thank Professor Dr. Mandana Madam for giving me the opportunity to talk to you about some of the work that we have been doing. Then many thanks to Oral Pathology India and Oral Path 360 Degrees. The kind of work that Madam has been doing over the past year has been simply exemplary, and trying to bring the entire dentistry together and the research and clinical aspects of dentistry in India on a global stage. So, what I'm going to talk to you today, it's always very daunting to speak to oral pathologists. And me being a periodontist is that see, generally what happens, people across the world, wherever I've seen, some of the great researchers, they are not dentists by profession, so they don't know the clinical aspects of dentistry. Dentists don't get into research because it's obviously not as lucrative as clinical practices. So oral pathologists, I'm always envy, because they are great, because they have their dentists as a background, and then they have exposure to research. So that gives them the cutting edge, or that gives them the advantage of being able to differentiate from statistically significant research and clinically significant research. So with that in mind, just want to present to you, coming back to the core, I want to discuss with you about the work that we've been doing over the last decade and a half on saliva. And I'm a periodontist, and I had the good fortune of completing my PhD in oral biology, which is predominantly microbiology and biochemistry to a certain extent. And my focus was on saliva in diagnostics and therapy. So although the topic seems to be a little dry, I'll try to keep it as energized and as interesting as possible. Okay, so why is saliva that important? Is because just to go run through some statistics for you guys. Periodontal disease approximately costs about $54 billion annually in lost productivity time. So that's the extent to which periodontal disease, gum diseases, and overall oral diseases cause to the exchequer. Diabetes, is expected, the cost is expected to reach about $2.1 trillion. And the same can be said about cardiac conditions. So why I'm saying all these things is, the reason why they cost a lot of this money is because they are not diagnosed early enough. And the reason why they are not diagnosed early enough is because either the costs of diagnosing is high, or it requires a lot of effort, or it's invasive to do that. Whereas when you look at saliva, it's a very non-invasive form of collection. So earlier non-invasive diagnosis, if it can be made, can significantly reduce the economic burden and mortality associated with these diseases. Now, in the words of Erwin Mandel, for those of you who recognize him as one of the doyens of microbiology, why saliva is such an important fluid when it comes to either diagnosis or to some even therapeutic case, is he very classically says saliva doesn't have the drama of blood. It may not have the sincerity of sweat, or may not even have the emotional appeal of tears, but still it's a very underrated body liquid, which does not get its due. So obviously, saliva being easy and non-invasive to collect, and the storage being quite easy, and you just have to spit. So volume is not an issue. You can ask people to spit how much ever they want. And especially in India, though we know, spitting is definitely not a problem, and it doesn't clot like blood, and so it's easy to manipulate. It's relatively safer than serum as far as exposure to bloodborne pathogens is concerned. Now, of course, we have to be a little bit more careful with COVID around. So apart from that, the advantage of saliva as a fluid, both used for diagnosis as well as therapy, is really great. Now, having said that, just want to, before I get into what is my actual research and how saliva has been, what we have been trying to use saliva for, just to give broadly what are the research trends that have been there with saliva. Is obviously, we know saliva has been used for a lot of diagnostic purposes, and of late we have a lot of microfluidics, and there's a company that manufactures something called as OFNASET, it's oral fluid nanometric assessment test, which basically just deals with a little bit of saliva that goes into a sample box and then gives you markers for a multiple variety of diseases and conditions that can include multiple varieties of cancer. You have arthritis, you have diabetic biomarkers, you have cardiovascular markers, and multiple infections, both local and systemic. So all possible with saliva. So a lot of focus is now being made from the diagnostic area into microfluidics. The second area of trend or research trend in saliva is predictive. So it tries to predict whether the person is susceptible to disease and not. And we have, obviously we have with the next generation sequencing, oral microbiome screening. And I don't know if any of you had the opportunity to listen to Purnima at the national conference in Perio. She spoke about the oral microbiome, and she's written some excellent articles written there. And now we have so many new emerging putative pathogens that have come up in saliva, both in terms of dental caries as well as periodontal disease, that needs to be addressed. So the salivary research is focusing even on predictive aspects, both in terms of microbiome screening as well as genetic sequencing of bacteria, and also multiple biomarkers. We've seen thousands, and the list is enormous about the biomarkers. Of course, the question always that we ask is whether it's a cause or an effect. So that question will continue to be there. And of late, and something that I'm very interested in is use of saliva as a therapy. And I wish I could speak to you, I can speak for hours on end on how we are now using saliva as a therapeutic agent for a lot of diseases, and even certain conditions which I'm not at liberty to disclose right now. But there are a lot of research that is going on currently worldwide on the therapeutic aspects. But one of the most important reasons why the research trend in saliva is now shifting towards a therapeutic is because of translational research. So we have seen many articles and we have seen many theses, right, about marker A, marker X, Y, Z is elevated in a certain disease if it's reduced in certain disease, and so on and so forth. There are thousands, but that really doesn't show us the whole picture. But as a translational research is the one that can actually show us give us a complete picture of how things are. So what exactly you translate or a translational research does is you start with a clinical problem. For example, a particular person gets a disease versus somebody who doesn't get disease, or a certain antibiotic shows a lot of antimicrobial resistance, is seen. So this is a clinical problem. So you always, translational research starts with a clinical problem. Let's say this particular person has developed, for example, cancer, or this particular person has developed a disease XYZ. And why is this person getting this disease versus someone who's not? And then you form a hypothesis. And you take this hypothesis and you take it to the lab. And in the lab, you test this hypothesis. And you say that, okay, there is a particular reason why this disease occurred in this person, or this drug does not act in this such person, or something. You form your hypothesis. You test that hypothesis. And then once you test it, you will fail. And then you do multiple tests, and then you fine tune it, and then you rework on your hypothesis, and you keep going back and forth between your hypothesis and your test. And ultimately, you will end up with some result from your test that will give you a little more holistic picture on why or how does the entire problem play itself out in the real world. And then you take that results of those tests and form some sort of therapy, and then you use that back into the clinics, and then that should solve the particular problem. So it's like a complete circle. Any research should follow this circle. It's not just that you hypothesize something and you do a particular research and you say that yes, there is some statistically significant difference between A and B, group A and group B, and so then yes. So that really does not give you the entire picture. Whereas translational research is really focused on getting you results, and that's why therapeutic approaches with salivary research is really taking off. Now, having said that, just getting back to saliva and our core area of interest, we know that saliva has got multiple areas. Since I'm addressing all our oral pathologists here, so I don't want to delve into the details of each of this, but we know that saliva plays a huge role in all of these functions, in digestion, lubrication, buffering. It's got a very strong buffering system, obviously the taste, maintenance of mucosal integrity. And what I'm really focused on and what has really appealed to me was the antimicrobial function of saliva. Now, years ago, I mean, almost 20 odd years back, when I was doing my post-graduation, I had the fortune or misfortune, I don't know, but I used to have a lot of cases of people suffering from aggressive forms of periodontal disease. So, and what I always used to find is that these people, they show very good response to treatment. So it's just like any other form of periodontal disease, whether it's chronic periodontal disease or anything. When you treat these patients, they show you very good results. But somehow, after 6 months, 8 months, when you recall these patients back, there is a recurrence of this disease. And I always used to think why this happens, and I somehow just left it. Now, just to give you a brief background on that, this disease that we earlier used to call localized aggressive periodontitis, I still feel is a distinct clinical entity which is not sufficiently addressed even today. Is caused by this fantastic looking bacteria, Aggregatibacter actinomycetum comitans, causes this particularly severe form of disease. And it's got this beautiful star shape when it grows in the wild. Why I say in the wild is because these bacteria exist in two different forms, and we call that as a rough strain versus a smooth strain. So normally in the wild, what happens is these bacteria fight, right? The bacteria fight with other bacteria to survive, survival of the fittest. So they try to exhibit all their virulence properties just so that they can cling on to the surface and kill the other bacteria associated and establish their dominance there. And bacteria in the wild are called as a rough type, and they exhibit this internal star-shaped structure as you see in this colony here. But when you keep growing these bacteria in the lab with a lot of nutrition and stuff, these bacteria feel they don't necessarily have to exhibit all these virulence factors to grow because everything is there and there's no competition to grow. So in such situations, they exhibit a different phenotype, and we call that as a smooth phenotype, which really does not auto-aggregate, or does not really stick onto surfaces, and does not have the star-shaped structure and stuff. But in any case, this is a fascinating bacteria that's implicated in the etiology of localized aggressive periodontitis. And this disease, as we know, is characterized by exorbitant amount of periodontal tissue destruction with hardly any local factors that is present, and typically affecting the first molars and central incisors. And also the way that we biochemically test this is by presence of the serum antibody response, and it shows a very high response as compared to the generalized antibody response, where generalized aggressive periodontitis, where obviously there is some sort of systemic condition also which plays a role, and there the antibody response is much lower. So this led this kind of findings during my postgraduate time and later years led me into my PhD, and I had the fortune of working with some of the best minds in as far as periodontal research is concerned. And I came across this article way back by Professor Daniel Fine. It said that something, the saliva. So what he did in the study was basically he took the saliva of people who had AA, and when he tested it, they found that it's something in the saliva kills Streptococcus mutans, which is the karyogenic bacteria. So the you always see a person who has a lot of periodontal disease may not have caries. A person who has a lot of caries may not develop periodontal disease, and I've always observed this in clinical practice as well. Of course, I'm not talking about occlusal caries, because occlusal caries is a lot of diet related in your your hygiene related, but smooth surface caries. So smooth surface caries, interdental caries, these are not generally is something not that you would find with people having periodontal disease. And so when we found this kind of distinct difference, Dr. Fine and his group at the lab did this study where he found that people who had AA in their mouths, they also had something in their saliva that was killing strep mutans, and they did this. So this is the they took the saliva, and if you look at the x-axis is a time of incubation, and y-axis is a log of bacteria. And so there was AA negative and AA positive subjects. So we had AA negative subjects. You could see the amount of Streptococcus mutans, which is the bacteria, reduced considerably, or almost you get a 2-log reduction over 45, 50, or 40, 48 hours or 50 hours of incubation. One log reduction is roughly 90% of bacteria is killed. So when you have 2 log, it means that 99% of the bacteria of strep mutans was killed by the saliva. And so this guy, and so you obviously went back and then started looking why this happens, and you find that obviously saliva is not a very simple liquid, right? So it's a complex secretion, and there are saliva can come from many different sources in the mouth, from major and minor salivary glands and stuff, so forth and so forth. And the evidence of antimicrobial function of saliva, we know, comes from a lot of studies, a lot of clinical findings that we find that in the absence of saliva, either patients who have oral cancer and they have undergone radiation treatment where the salivary glands are being affected, you see that there's a lot of radiation caries and stuff that's involved. So you obviously come to know that saliva does play a critical role in antimicrobial function, both in terms of lubrication, in terms of washing away of these pathogens, and more importantly, also in killing certain pathogenic bacteria. So when I got this idea, I saw this article, and then I said I wanted to do something for AA, because I find that I was more interested in aggressive periodontitis. So when I joined the lab, we started working on finding out what is it that AA has got to do in saliva. So what I did was we took a saliva from 10 different volunteers, and we found we tested those saliva, and we tested the antibacterial effect of the saliva against AA. So here's what this graph. So the x-axis is all the subjects from 1 to 10, and your extreme left is just the control. And on your y-axis is the log of CFU. So what it says is the bars represent the AA bacteria. So higher the bar, greater the bacteria; lower the bar, lower the bacteria. So when we tested AA against saliva, we started with equal amount of bacteria in all the tubes, and we took the saliva of 10 different people, and then we incubated it for 2 hours. And after 2 hours, we tested how much amount of AA is present, viable AA is present. And we found that 2 subjects, subject 3 and subject 6, had something in their saliva that was significantly reducing the amount of AA. So, and that led me to kind of conclude that there is something in the saliva of some people who don't have AA in their mouth that actually kills AA. So this was a great finding for me, because that answered something, that saliva there is some component in the saliva that is actually killing aggregated Bacteroactinomycetum comitans. So in order to find out, so what happens is saliva, like I said, is a complex fluid, right? So you have proteins, you have, other than proteins, you have so many other enzymes, and which are all proteins. You have non-proteinaceous things as well that can exhibit antimicrobial effect. So I wanted to check whether this factor in the saliva that is killing AA, whether it is a protein or it's not a protein. And the way that you find that is you take the saliva of these people and treat it with a particular, an enzyme called proteinase K. Proteinase K is an enzyme that actually chops up all the proteins. So the idea was, if the proteinase K is chopping up all the proteins, then the protein in the saliva, which is killing the AA, would also be killed by this proteinase K. So effectively, if you treat the saliva with proteinase K, you should lose all the antimicrobial effect, and that's what happened. So what happened is when you took the subject 6, I took the saliva from subject 6, and then I checked the level of with AA. You can see here that there was a reduction. There was a 1 log reduction, which is roughly about 90% of AA was killed. But when you treat it with proteinase K, it kind of, the same saliva lost the killing efficiency, which meant that the component in the saliva that was killing AA was actually a protein. So now that was another breakthrough for me, then thinking that yes, something in the saliva which is killing AA is actually a protein. Now, the idea is, so there were, thousands of proteins in the saliva. So how do you know which protein in the saliva is it that is killing AA? And I was discussing this with my mentor, Professor Fine, and he said, anything that kills has to bind first. So obviously, the bacteria, any protein in the saliva killing bacteria first has to bind to the bacteria, and then it has to kill it. So by using a technique called as affinity chromatography, we could isolate proteins in the saliva that was sticking on to AA. So here's what we did. I don't want to explain the slide, but basically what we did was we packed a column, maybe the next slide. Yeah. So if you look at this top left here, you can see that there is a tube with all containing different proteins in the saliva. And this column was packed with AA. So what then happened was we passed saliva through this column containing AA in it, and whatever proteins in the saliva that had effect against AA would stick on into the tube, and which proteins that did not bind to A would come out, right? So we loaded this affinity column, and then we kept on washing it till no further proteins came out. So that is, all the proteins which were there in the column was basically stuck to the bacteria. And then using eluting, we started eluting those proteins out by adding dilute HCl. And then we started eluting those proteins in the saliva that were sticking to AA. And then what we did was we ran a gel. Obviously, when you run a gel, you can identify all the different proteins. So as you can see on your right, you have different proteins on your SDS page analysis that you find are stuck in the column. Now, all those lines that you see depict each of those proteins, and the names of those proteins are present there. But there's one particular protein at the bottom. There's a very huge band, thick band, which shows there's a huge amount of protein that has actually come out. So we thought that, since that protein shows the maximum amount of concentration, that means it has high affinity for AA. So when we cut that band out and we sent it for sequencing to find out through MALDI-TOF, we came to know that this particular protein is cystatin. And so now, so far, what we know is that cystatin SA is a protein in the saliva that is binding to AA, and we also think that it could have an antimicrobial effect against AA. So far, that's the story. So when we collected these fractions out, we wanted to purify. These has a bunch of proteins in it. So we just to purify this cystatin SA, we used a molecular weight filter. So there was, cystatin is roughly around 14 kilodaltons in molecular weight. So when we did a 15 kilodalton molecular weight cutoff filter, what it means is that anything that is above 15 kilodaltons in weight will get stuck in the filter, and everything that is below 15 kilodaltons would come out. So it's like that, like a coffee filter, So once we did that, and we actually got pretty pure. Sample purity was again confirmed by using a Western blot analysis using antibodies against cystatin SA. And that's how we found, and that's how we purified. So this step was purification of cystatin SA from the column which was showing high binding towards SA. So now we have pure cystatin SA, which we think could have antimicrobial effect against AA. So just to give a background now about what cystatins are. Basically, cystatins are nothing but inhibitors of cysteine proteases. So there are three families of cystatins, family 1, 2, and 3. Don't want to get into the details, but SA stands for salivary cystatin. So that's why the name came salivary S. When you have salivary S, SA, and SN, which is acidic and neutral and so on and so forth. So basically, they are just isoforms of each other. Now, the cysteine protein, what are cystatins? It's an inhibitor, right? It's an inhibitor of proteases. So proteases are a group of proteolytic enzymes that will cleave, they'll chop up the proteins at peptide bonds, and the CONH bonds get broken down. And there are many examples of proteases like endoproteases, exoproteases. These are produced both by the host as well as by the bacteria. And these proteases, if you leave it uncontrolled, they will lead to a lot of tissue destruction. So basically, cystatins are nothing but inhibitors of cysteine proteases, which again help in controlling inflammation. But these are nothing to do with antibacterial effects, right? So far, we did not know whether cystatin that we have purified, there were not many published reports on whether cystatins have got any antimicrobial effects. Of course, there are some peptides that are derived from cystatin that are shown to have antimicrobial. Some evidence came from some researchers back in 1996, both for A as well as P. gingivalis. No, not for A, but for P. gingivalis. And they also concluded that, in that study, they concluded that the mechanism of inhibition was independent of protease inhibitors. So the idea is cystatins being protease inhibitors will prevent the bacteria from breaking down the proteins, thereby depriving the bacteria of nutrition, and that's how the bacteria would die. So that was what a possible hypothesis was for cystatin. And there were some other studies which said that peptides, synthetic peptides which are made out of the similar sequence of cystatin was shown to have antimicrobial effects. So based on our data and previous studies, we hypothesized that cystatin SA might demonstrate antimicrobial effect against aggregated Bacteremia by stem chromatins. So the story so far is that I found certain saliva of some people kills AA, and then we took the saliva of these people and we purified the saliva, and we found that this saliva kills AA. We also treated that with proteinase to know whether the thing that is in the saliva that is killing AA is a protein or not, and hence we found out that it is a protein. We then ran an affinity chromatography to make sure that the proteins that are killing AA will also bind to it with the hypothesis. And so we got this set of proteins which were binding to AA in the affinity chromatography column. From there, we purified cystatin SA, and now we have pure cystatin SA with us. And now we want to check whether the cystatin SA specifically has an antimicrobial effect against AA. So with this, we tested cystatin SA against not only AA, but a few other pathogens, which included Fusobacterium nucleatum, strep mutans, and Staph epidermidis. These are nasty bugs. We can go. So here's what I found. So this is a typical agar diffuse assay. We found that on your right is saline. So when you treat it with saline, obviously there's no inhibition, there's no zone of inhibition that you see. But when you treat it with this purified cystatin assay, in a filter disc assay, we found a clear zone of inhibition that was found around cystatin assay, which again led us to believe that yes, there is some sort of inhibition of AA. Now, whether this inhibition is actually killing or it is just preventing the bacteria from growing, we don't know, right? So we did a killing assay. So the next what we did a AA killing assay just to check whether cystatin actually kills AA. So we tested it on multiple serotypes. So you know that as of now, we have 6 different serotypes that have been identified. IDH781 was a serotype D, which is the clinical strain, a rough isolate. and which we commonly use to check because it's very closely resembles the wild type strain. And then we tested it against other pathogens, other serotypes, B, C, and so here you have in 3 different concentrations. So if you look at the green arrow here, there are 3 different concentrations of cystatin. The darkest green is the 0.1 milligram. The second, the lighter one, is 0.05, and then the third is 0.01, and the gray bar is just PBS. So this is a control. So all the experiments obviously performed in triplicate and duplicate in at least 3 different occasions. So here's what we found. There was a dose dependent decrease in killing activity. So 0.1 milligram. We also did a 0.5. I'm not showing that results because 0.5 and 0.1 was similar, and that's how we decided to make it a 0.1. So cystatin at 0.1 milligram showed almost a 1.5 log, 1.2 to 1.5 log reduction, which is roughly 95% of the bacteria of A that was actually killed. And this effect was replicated across all the different serotypes of A that we tested. We also tested it at different time points. We tested it for 30 minutes, 2 hours, and 4 hours. So we found that at 30 minutes, there was not sufficient killing, that it required at least 2 hours to demonstrate its effect. And exposing it for longer period did not make any further difference in its killing activity. And then what we did also is we took cystatin SA antibodies. Sometimes there could be a contaminant in your, purification, or there could be some other salt in your sample that could have killed A. So what we did was we took particularly cystatin SA antibodies. We added the cystatin SA antibodies to cystatin SA to absolutely negate cystatin SA. And then we found that as you keep adding the antibodies, the antimicrobial effect also reduced, which again confirmed that the cystatin SA was actually killing AA. But again, just to reconfirm whether it is killing, we wanted to visualize whether it kills or it's just not growing or it doesn't kill. So we did a confocal microscopy. So obviously red means it's all dead, and green means it's all alive, and orange means there's some alive and then some dead. So when you look at this picture here, when you treat it with the top row is cystatin, and the bottom row is saline, and ABC is different concentrations of cystatin. So when you look at cystatin SA at 0.1 milligram, you can see a sea of red, which means that the cells which that is the AA cells, the AA colonies and AA cells that were exposed to 0.1 milligram per ml of cystatin SA were all red, which means they were all dead. They were all dead bacteria. So obviously, this is a very clear indication here that cystatin SA demonstrated significant anti. So we published that. And now when we checked it against other bacteria, we found it was not effective against F nucleatum. So in the top left corner in the blue graph, you see that there was no difference in the bacteria, the Infusibacterium nucleatum. So cystatin SA was ineffective in killing F nucleatum. It was showed a marginal amount of killing against both Strep mutans and Staph epidermidis. But it showed quite a lot of antibacterial effect against Porphyromonas gingivalis. So here's what we found. We found that cystatin SA binds very avidly with a lot of affinity to AA, and it also exhibits killing. And this killing is a dose-dependent killing, and as evidenced by confocal, it's not just inhibition; it also kills the bacteria. Just to reconfirm, we also treated thing with a loop with pre-mixed the cystatin, the purified cystatin with lupeptin. So what lupeptin does is it inhibits the protease inhibitory function. So once you treat it with, it's like basically like antibodies of cystatin. So once you treat it with lupeptin, it will. So we wanted to know whether the effect of the the killing effect of cystatin SA, is it related to the protease inhibitory function, or is it related to so every protein will have certain domains, right? So you have sequences, the N terminal at the C terminal, and basically the structure of the protein is what determines the function of the protein. So we know that cystatin was a cysteine protease inhibitor. So whether the killing was related to the protease inhibitory function, or was it independent of the protease inhibitory function? So what we did was we took another protease inhibitor, which is called as lupeptin, and when we checked AA against lupeptin, we found that lupeptin does not kill AA, but cystatin does. So lupeptin and cystatin SA have very common functions, that they both inhibit cysteine proteases. So if the killing of cystatin was related to the protease inhibitory function, then lupeptin also would have killed AA. But lupeptin did not kill AA, but cystatin did, which showed us that the antimicrobial effect of cystatin is not related to the protease inhibitory function, right? So this was part one. So now we got to thinking. So we published this part, and we concluded that yes, there is something in the saliva that kills AA, and this something was cystatin, and this cystatin was actually killing bacteria, especially AA, and to a large extent even P. gingivalis, but did not show much effect against strep mutans, staph epidermidis, and F. nucleatum. And that we know that cystatin SA, the antimicrobial effect was not related to the protease inhibitory function, and it was dose dependent, and it was time dependent, and the concentration at which it killed was 0.1 milligrams per ml. So these were the conclusions that were derived from this first set of studies. Now, from this, we moved on to the second part, is basically how does cystatin kill? So we know that it's not related to the cysteine protease effect. So how does it kill? So what I set out doing was, the initial hypothesis was any protein that has to kill has to bind. So then it got us to think, like, we know that cystatin binds to AA, but what part in the AA does it bind to? So A is a gram-negative bacteria. So obviously we did an outer membrane preparation, and we took out all the proteins in the outer membrane of AA, and then we ran it onto a column. I ran it onto a gel. And then what we did was we treated that gel, we transferred that gel to a blot, Western blot, and then we incubated. So this is how say the outer membrane proteins of the AA looks like on the gel. And then what we did was we transferred that to a Western blot. In the Western blot, we immersed the Western blot in cystatin. So the idea was cystatin would bind to some of the proteins in the outer membrane. So now that, to identify which proteins it's binding to, we soaked it with cystatin for overnight and washed away all the cystatin off. So now whatever is remaining is the cystatin that is bound to the protein in the gel. And then we used anti-cystatin antibodies, and then we used immunofluorescence to find out that there were actually two proteins on the outer membrane of bacteria of AA to which cystatin was binding. So the hypothesis now, as we said, was the cystatin binds to A, and how it binds to A is through these two proteins. So what are these two proteins? We send these out for MALDI-TOF sequencing, and this is the actual photograph. And what we found was there are two major proteins to which A binds to. Cystatin binds to on A, that is OMP39 and OMP34. OMP is nothing but outer membrane protein, and 39 is just the molecular weight. So OMP39 and OMP34 are the two proteins to which cystatin binds to on the surface of A. So just to give you a little bit of idea on what OMP39 is. So when you do a blast is it's a bioinformatics tool. You're trying to find out what exactly it does, what is the structure and function, and the same thing in other relevant bacteria and species. So we found that OMP39 is very close to OMP-C protein seen in other bacteria like Mannheimia. So what OMP39 is also been known for, it's a porin protein. So porins are basically like channel proteins through which it allows the diffusion of nutrients and fluids. We also found that bacteria can also, like how we get attacked by viruses, bacteria also gets attacked by phages. So this OMP39 is also was known to be a major site for bacteriophage attack in other species. And they've also been shown this outer membrane protein 39 to be involved with attachment. So like I said, for every bacteria to exhibit disease, the bacteria first has to colonize. So this OMP39 has been implicated in attachment. It's also been shown to be a major site for bacteriophage attacks. And it is also similar to most porins. And they are actually used as candidates for vaccinations. And OMP34, we had some data, especially these are some studies that come out of Mark Wilson's paper back in 1991, which showed that the serum or the sera of patients who had this localized aggressive periodontitis had showed very high amount of antibodies to OMP34. So you see what's happening here. OMP34 is present on AA. Now your body recognizes AA by certain antibodies. And these antibodies are targeted against proteins on the AA, which is your OMP34. So OMP34, to me, when I was seeing that, it showed that as a potential candidate against which targeted therapies can be done to prevent colonization of AA in the mouth and to prevent it from causing disease. Some of the other functions that were associated with OMP34 that we found is shown to be attached to involved in bacterial adhesion and invasion into epithelial cells. So to make a long story short, what we hypothesized is that cystatin SA is binding to AA through OMP39 and OMP34, and then we hypothesized that OMP39 and OMP34 is the mode of entry of cystatin into AA, and thereby it kills AA through this OMP39 and OMP34. So how do you prove that? So the next step was I took the gene pool of AA and we cut out OMP34 gene, and we inserted this OMP34 gene into another bacteria through a vector, the 2.1 topo vector, standardly available for proteins and other things. We took this gene and we put it into E. coli. Now, normally cystatin does not kill E. coli. So here's what we found. So when we call a gene put a gene into another bacteria and try to stimulate protein production, we call that as recombinant. So what happens is normally in the wild type, which is a DH5 alpha, the DH5 alpha is a wild type E. coli. When you treat it with cystatin, no matter how much your concentration is, it does not kill E. coli at all. But then you have this recombinant E. coli, which is now having the OMP34 and the OMP39 expressing on its surface. Then you see that cystatin has no effect on OMP39. So if you look at the top left, when the E. coli was expressing OMP39 via plasmid PGK1, there was no killing of the AA or of the E. coli expressing OMP39 by cystatin. But when the E. coli was expressing OMP34, with the wild type, which is not being killed, suddenly became susceptible to attacks by cystatin. What it showed to me by these studies was cystatin kills AA, and the way that it kills AA is it binds to OMP34, and it uses OMP34 to get inside the AA cell and then breaks it down and breaks it up. So this was what we studied with the help of some genetic assays and some of the genetic stuff. So we also tried to create, so the idea was, now you take A, now A is having OMP34. Now if you create a genetic mutant of A where you knock out OMP34 gene, so that the A, which is getting killed by cystatin, is if you knock out the gene, the A should now become resistant right? The A should become resistant to cystatin. So then we set about creating a genetic knockout of AMP34 in A. And trust me, I spent close to 6 months on this, and I could not create a genetic knockout. We did create some of the other gene knockouts like AAE, which is another protein, outer membrane protein in the on the surface of AA, which is involved in attachment and adhesion. But we could not create OM34 knockout, which probably shows that OM34 is a very critical gene. So if you mutate that gene, the bacteria does not survive. So we had to fail back on that. So when we checked again, we couldn't knock out the gene in AA, but we could express AMP34 in the mutant AMP34 in E. coli. So now you have E. coli, which normally does not get killed by SA, cystatin SA, and now it's expressing AMP34, the wild type AMP34, and it's killing. Now E. coli, which is not getting killed, is now getting killed by cystatin when it's expressing AMP34 on its surface. And when we reintroduced the mutant AMP34 back into the E. coli, now again this E. coli became resistant again. So it shows that cystatin SA was killing the bacteria using OMP34, and when the OMP34 was mutated, then the cystatin lost its antimicrobial effect. So this was convincing enough proof that A, cystatin SA was showing antimicrobial effect against A. Number two, it was independent of its protease inhibitory function. The way that it killed was binding to OMP34 and OMP39, and expression of OMP34 in E. coli cells made E. coli, which was otherwise resistant, susceptible to killing by cystatin SA. So this second set of experiments, which lasted about a year or 1.5 years, led us to some of these conclusions: is that cystatin SA binds to OMP34 and OMP39 on the outer surface of A. It also exhibits antimicrobial effect against E. coli expressing OMP34. No effect was seen on E. coli expressing OMP39, so which means that it binds to OMP39, but it does not kill by OMP39. And the antimicrobial effect is reversed when the OMP34 gene is mutated. Now, with this in mind, we also did, obviously in the, these are all labs. So like I said, when you talk about translational research, what we have to understand is whatever hypothesis that we formulate in the lab has to be clinically driven and has to be clinically relevant. If you look at some of the articles that are published, now, normally in the pathogenesis of periodontal disease process or in any disease, the first step in disease production is colonization. So when we look at even periodontal disease, we looked at especially aggressive forms of periodontal disease, and we look at the primary pathogen, that is AA. We looked at where AA colonizes in the mouth. And what we realized is the reservoir in the mouth lies in buccal cells. So imagine what happens in the mouth. A is actually present in the buccal cells because A is a very poor colonizer. A cannot is not very great at colonizing. What it does, it stays inside buccal cells. It attaches to buccal cells, it invades buccal cells, and it waits for an opportunity to enter the subgingival environment from the buccal cells through sloughing off of buccal cells. So what then happens is in the subgingival environment, you have your other putative pathogens and so many of the other pathogens. But when AA finds an opportune moment to move from the buccal cells into the subgingival environment, and then it works. And there are multiple studies which show that buccal epithelial cells is a primary source of attachment. And so not only does AA bind to buccal epithelial cells, it actually invades buccal epithelial cells and stays inside the buccal epithelial cells. So when I tested cystatin SA and on AA, and so if you remember, I tested 3 different concentrations. So when I checked cystatin SA in 3 different concentrations and tested the ability of the AA to bind to buccal epithelial cells, what I found was the CFU per BEC, that is the number of bacteria that bind to every buccal epithelial cell. At a 0 0.1 there was hardly any, 0.05 there was hardly any, but 0.01 we still got about 80 bacteria that bound to the buccal cell as compared to 140 bacteria per buccal epithelial cell. Now what this shows is that cystatin decreases the binding of AA to the buccal epithelial cells. You obviously cannot correlate this data with 0.1 and 0.05 is probably because they were killing the bacteria. But 0.01 dosage does not really kill the bacteria when we checked before. So at a sub-antimicrobial concentration of cystatin, which when we treat the AA, pre-treat the AA with the sub-antimicrobial dose of cystatin SA, it also prevents the binding of cystatin SA, of binding of AA onto the buccal epithelial cell. So we don't want to get into these, but what it basically means is that you have cystatin SA, which interferes with the ability of AA to bind to buccal epithelial cells. And basically the way that obviously think is OMP30, cystatin binds to OMP34 in the AA, and OMP34 is essential for binding to buccal epithelial cells. So cystatin via its subantimicrobial effect is binding to OMP34 and OMP39, which are basically your attachment sites, which exhibit binding to buccal epithelial cells. And finally, we had to conclude that, to get to the loop, to the clinicals. So what we did was we wanted to know, these people who have aggressive periodontitis, whether there is any difference in the levels of cystatin. And there were some previous reports which showed higher, some previous reports which showed lower, so on and so forth. So what we did was we had an ongoing study on a long-term study of high school kids and adolescents showing localized aggressive, because generally African Americans are generally more affected by this. So we had a long-term study seeing these children, and we took the salivary samples and checked for the concentrations of cystatin. So when we looked at people who are not affected by this disease, the concentration was close to about 300 micrograms per mil. But when you look at people who had this disease, their concentration of cystatin was almost 60% reduced. So this kind of completed the entire loop and showed that cystatin binds avidly to A, and this newly discovered property might be easy. Now imagine therapeutically what you can do. So you can take, you can find out which part of cystatin SA protein is actually binding to AA and create a synthetic peptide which can actually block the binding of AA to the buccal epithelial cell. You can use it for many of the others, including chronic periodontitis, because it shows a lot of effect against P. gingivalis as well. So we can also, it also showed cystatin SA demonstrates antimicrobial effect against AA, and this effect might be bactericidal in nature. It's not just an inhibitory growth. And then cystatin, we know that binds to OMP34 and OMP39, which can be used, like we said, as a potential vaccination and stuff. And then antimicrobial mechanism of cystatin SA is mediated through this outer membrane protein 34. So these are some of the findings associated with my study, and cystatin, so the levels of cystatin SA was decreased in patients with periodontal disease. So in future, what we have to do is, now we found out so we've published about 3 articles in between all this work, some in JP, some in Journal of Periodontal Research, some in Infection and Immunity, Journal of Antimicrobial Agents and Chemotherapy. So we've published a few of this work. We still have to publish the entire picture, and still is going on, but we do have a provisional patent on this. So some of the future directions that because we do know that cystatin SA has got some polymorphisms associated with it. So we want to check whether the different polymorphisms of cystatin SA does affect the function, the antimicrobial function of cystatin SA as well. Now we also want to know what is the receptor on the buccal epithelial cell. for the buccal for OMP34, so that we can create a decoy receptor also. So if we create soluble decoy receptors, we can also put that in a toothpaste or a mouthwash formulation or whatever, and that, especially in susceptible population, can help in reducing the colonization of these periodontopathogens in the sulcus or into the buccal epithelial cells. All right, so these were some of the thing, and I would like to at this point acknowledge all the people, my mentor, Dr. Daniel Fine, Dr. Kabilan, who worked with me, taught me a lot of the stuff, the techniques and associated. Dr. David, God bless him, rest in peace. Dr. Helen, Professor Mari Basappa, who's also an expert today. I thank him for his valuable contribution. Dr. Sendil helped me a lot with those genetic assays. Debbie, sweetheart, she always supported me through this entire process. Valerie was there, Carmen, Miriam, Marie, Amy, some of my students, Prerna, Vandana, Deepthi, and also the staff and faculty at the college that I work currently, my dean, Dr. Sunal Mudaya, who supported me a lot during this career, during this research, our principal, Dr. K.C. Ponappa, my fellow colleagues at the Perio department, Dr. Amit Walvekar, Dr. Jagdish Pai, Dr. Annaji Sridhar, Dr. Sachin Malgi, Dr. Radhika, my PG students, Dr. Gopika being one of them. She did some of the work with which we have not shown actually, but different types of serotypes in healthy, chronic, and aggressive forms of periodontitis. It's a very fascinating study. So thanks to one and all, and finally, thank you, Mandana Madam, for inviting me. And I hope I didn't bore you guys with all this. And I'll be ready and free to take any questions at this point that you have. Thank you.
Yes, that was a great presentation, and I can now see that I misplaced it into the dentistry special. It would have probably been better placed into the research special. So, okay, I think I have Dr. Krishnaraj. Are you there with us? Can you put on your mic and your.
Ma'am, I'm here. Yes.
Okay. Can you turn on your camera or not possible?
Yeah, I can, but due to network issues, I feel there may be a lag. Let me try putting it down.
Okay, just see if it's possible. Okay, all right. Well, as everyone can see, he is driving somewhere on the way. So, but he can answer the questions. And I think we will, Dr. Maribasappa seems to be off for a few seconds. But we have the first question. I will bring it on the screen for you. So it is superb study. It may be nucleus for many future immune therapies. It's a comment from Dr. Nasser from the USA. And I can also request everyone else to please start posting their questions and comments so we can share them here. Now, I have my own question, so let me start with that. At what actual stage of clinical use are we with any of this?
Well, it's a great question, ma'am. Like with most therapeutically driven strategies, there is a lot of distance between cup and lip. So it has to go through that different stagings, different processes, and getting through the patent, and then finally getting into therapy would mean somebody has to buy it, and somebody has to think that it's worth actually investing a lot of money to get it into the market. But yes, like cystatin, there are many others also which have shown tremendous promise. Whether cystatin alone will make a significant difference is something that we don't know yet. But we do know this, that we identified some new function with the protein. And, in biology, in systems, we know that many of these proteins associated in the mouth or anywhere in the body have moonlightings. So they have multiple functions. So some of them have not yet been discovered. So definitely this will lead a way or lead a path to trying to figure out many other proteins which may work synchronously to create the benefit. Yeah.
That is true. Okay, now that we have Dr. Maribasappa back, I think can you give us share your experiences and your thoughts now on whatever was covered so far?
Oh, yeah, sure. Yes, I have enjoyed Dr. Krishna. We used to, we worked in the same research lab in UMDNJ, which is now called as Rutgers School of Dental Medicine. So I know Raj very closely. Just one general comment, initially he made that the dentists are not so interested in research, and most of the research is done by non-dentists. I am also a non-dentist. I am a microbiologist doing research in oral microbiology. But I think I got in touch with Raj quite recently. I am not sure how active he is, but this problem he mentioned is also true for Raj, if that is the case. He was such a talented researcher. I could see him. We used to interact every day on a regular basis. We used to talk about the issues in research and troubleshooting and so on. Such a talented guy, but yeah, that was just a general comment. My being in oral microbiology research for about so when did I start? 2002. Yeah, about 16, 17 years. My view is that it's it's now the time, at least since last 7, 8 years, I would say, when the genomics and the next generation sequencing has become more affordable by researchers. It's quite heavily used. Many people use it for characterizing the oral samples, be it plaque samples or saliva related to oral infections. If we talk about localized aggressive periodontitis, which was the focus of today's talk, like in terms of oral infection, there was a time in late 90s and early 2000 that Aggregatibacter actinomycetum comitans was portrayed as a like monobacterial infection causative agent for localized aggressive periodontitis. Um, I could mention a couple of names who, like Morgan's Killian and Habeck. These people identified JP2 like strains. JP2 is like J and P stand for the name of the patient, and these type of subtypes of Aggregatibacter produce heavy amounts of leukotoxin, and they proposed that AA almost functions as a monobacterial infection, single causative agent for LAP and cause the disease. Later on, they actually have several studies demonstrating this, but other researchers have shown otherwise also, that it's not just AA, but there are other players. So my belief in this is that microbial ecology in the oral cavity is quite complex. It's quite intricate. And the same is true also for interspecies interactions. It's no more just one single molecule, one single species, and then you just hit it. It's not like that. I'm sure Dr. Raj agrees with this. Recent microbiota studies have revealed quite many new novel species. Novel in the sense, nothing is known about them except that their quantities, their abundance is either elevated in the oral samples. So what does it mean? Is it enough that they are in high numbers? That is not known. I could mention a few of them, like Filifactor alosis and Leptotrichia shahi. There are quite many such species that are elevated in oral infections. So much needs to be done. There is a lot of scope for research, oral microbiology research. Young researchers can take this up and then do similar things like what Dr. Raj did several years ago. Because the question has to be addressed individually. Again, when I said it's no longer one species and one single molecule, but everything has to be put together. You have to study, of course, in a dissected way. You have to look at individually and then get a comprehensive view of this. So this is my idea. So microbiota and microbiome in oral microbiology is the current trend since at least last 10 years. So much has been going on. Yeah, so role of these novel species could be studied, and there is so much to know still. There was one, this was like my general comment, but I noticed one thing which might be unclear to some of the audience. I'm sorry. In one of the slides Raj presented, where OMP was it 39 or 34 was expressed in E. coli, mutated form of OMP34. When it was expressed in E. coli, it was not, it did not possess, it did not demonstrate any antibacterial effect. That, I think he meant probably truncated, Raj. It was not a full form of OMP34, but probably you took
Yeah.
out a domain.
We took out
Right?
the N
Yeah.
terminal. We took out the
Yeah, domain. So N-terminal domain was shown to be possessing the antimicrobial effect.
Yes.
Yeah. If there are any questions relevant to oral microbiology, oral microbiota, microbiome, and salivary research, we could take up.
There are so far no more questions. You know what I have experienced now in the recent past while doing this is that when there are no questions, that itself tells me that this is a topic that we are sort of ignoring. And there's not that much activity happening on the topic, and therefore there is not that much. Everyone is listening in, and
Yeah.
hopefully many will go on to, work with it. But the problem is we are not really doing enough. So therefore, we don't really have that much questions. And I think one of the very good things you said, that there is a change in the oral microbiome, and I think the reason that dentists would be very well placed to study this. Of course, basic sciences like you are contributing much more than we are as dentists at the moment. And really, oral microbiology falls in the forte of oral pathology. So, me and my colleagues are the most responsible for that not happening. So, we should take that blame. But the point is, that everything we as dentists do will inevitably change the oral microbiota. We place a restoration. Depending on what kind of restoration, we place an implant. We even do a scaling for that matter, which is good because we're obviously removing all the tartar and all the calculus. But every one of those is going to, in some way, I believe, and you can correct me on this, is going to again further change the oral microbiome, the overall presence of the microbes. So it is a very constantly changing and evolving situation. Very difficult to identify in that which are the normals and which are not, which are routine, I should say, not normal, which are the routine microbes and which are not. You can add to this now.
I agree, yes. But how persistent that kind of change would cause, to, for oral infections to manifest, dysbiosis is key. So if these kind of dental interventions are only temporary, causing not significant changes in the microbiota, that wouldn't be an issue.
Yeah, Mari, this point is very good because it has a lot of relevance to clinical.
Yeah.
Dysbiosis, when it occurs, and then when there is a temporary intervention, which means when you translate it to clinical practice, it means people don't go for regular follow-ups and regular checkups, and people don't get routine oral hygienes done. Then these issues may definitely develop. So,
Right.
when we talk about microbiology and the entire oral microbiome, and the complex interactions that we have with both local as well as systemic. So I guess it's time that the public health authorities take it up and include oral health or oral hygiene as a part of the general insurance, that they get provided, a couple of
Exactly. Yes.
hygiene
Mm.
treatments in a year, and routine follow-ups, and that should be mandatory. So I guess
Yeah. Yeah. Yeah.
that's that would be
Yes, I still remember, Raj, when we were in New Jersey, we have spoken about this issue
yes.
sometimes. Even today, oral health is so much ignored in India, like the common people. They just don't care. The no dental visits. They just think, no, it won't kill us. So that is the biggest problem. There is so much. I mean, connection has been established now between the oral infections and the systemic diseases. Not necessarily that oral bacteria just go and enter the cardia, cause heart problems there right away, but being located in the oral cavity, they can still do so much damage. Yeah, it's known, but I think Raj was telling at that time that there has been I don't remember very well, Raj, what was your response? That there was some action plan by the government, like to promote oral health. It was long time ago. I don't know. I don't see any
So there were a
much
couple of
progress.
insurance companies that started up, you know, allowing dental treatments,
Hmm.
but such as they could not have a regulatory approval or control on the quality aspects of it, and there were some other issues with use and misuse that that kind of, the government
Yeah.
and the companies rescinded on that. There are still a few companies which do allow certain sort of reimbursement on dental treatments and procedures, but nothing from the government yet on including dental treatment, especially basic treatments like hygiene should be included as a part of. If a person is insured for a medical insurance, he should be provided at least two hygiene in a year. They should be free for the patient.
Right.
I think
Yeah.
it should be.
Yeah, at one point I was even thinking, in India we have these packages or help that is given to the PBL families, so they get a certain quantum of their requirements at a subsidized rate or even free of their daily groceries and stuff like that. I really have been thinking and trying to get people to listen that we need to have a movement where that includes things like soap and toothbrush, toothpaste. I think they are now essentials. They are not luxury. They are essentials. And if people can't afford it, we should be able to really help them, because that then reduces so much additional later cost. I mean,
Yeah.
today we know that gut microbiota has been linked to cancer,
Yeah.
quite well established,
Good.
if I'm not mistaken. So, what
Yeah.
is to say? I mean, oral microbiome might as well be linked to a number of things which we are finding slowly, but it could. Prevention costs very little, but we
Yes.
need to move on that. Yes.
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 Yeah.
So, we have a question. Let me bring it on the screen. So, Dr. Nasser says, can you comment on steps against one second steps of relationship to autoimmunity, and how was your research help in further elaborate that? How does your research help in that? Further elaborate.
I didn't quite understand. Is it the
I think it is
relationship
basically
to
relationship
autoimmunity?
with autoimmunity and the microbiome.
Um, well, I'm really not the authority to answer on this. We have not done much of immune studies, but a little bit on Perio 2000, to quote, it's a Th1 Th2 response to presence of infections that can actually trigger some sort of immune mediated conditions that can persist in the body for a long time following an onset of periodontal disease that may contribute to tissue destruction. So that's a hypothesis, but really I have not done any research on that, so I'm not the authority to comment on how it can trigger autoimmunity.
No.
Okay.
Yeah. Yeah.
And then, of course, we have Dr. Sandhya, who says she's going to listen to this many, many times. because there's a lot of points in there that can be used, and I agree with her. And okay, I think Dr. Nasser meant with focal epithelial autoimmunity, depending on the immediate location. But anyway, again, we'll find that answer somewhere else. Um, and okay, yes. And that is again Dr. Sandhya saying it has clinical application. Yes, but we, the dentists, need to take it into the clinical side. It's very funny. I remember, and I don't know if this is still the case, but 30 years ago, when I was doing my BDS, we had to do every physiology, biochemistry, and pathology test that the medical students did, including testing urine. How useful?
Mm.
Well, never mind. But I don't know. We never collected saliva. We never tested saliva. Not even a salivary flow rate was a part of our BDS curriculum.
Yeah.
So I think we really treat saliva as the unwanted stepchild of dentistry. Everybody only thinks of saliva as too much during treatment or too little again during treatment. And that's it. And the rest of the time we sort of ignore it, sadly. So I'm not a clinician, so I'll let Dr. Krishnaraj comment on that.
No, ma'am. I mean, I wouldn't say. I mean, see, research. I remember, if you remember, Dr. Murray, we had a situation where there was. Don't want to name the company. That the company came up with a peptide saying that we got this smart peptide that's going to kill strep mutans. And they invested like $1 million on it, and they
Yeah.
got it to our lab. And Mari, me, Dr. Kavilan, and I think a few of us sat on it and said that, does it really work in the real world? So we took the saliva and mixed with this peptide and tested the effect on strep mutans, and it was zero. When when we tested the peptide independently, there was a 6 log reduction. But when we mixed it
Mm.
in saliva, they saw saliva has got multiple amount of proteases and stuff that can make any antimicrobial. So if it has to work in the real world, it has to work in the presence of saliva,
Yes.
especially the mouth.
Yeah. True.
So we realized that how dynamic saliva and working with saliva is, and the company realized it the hard way after losing $1 million on it. But yes, it is definitely a challenging environment to work in.
Yeah.
Yes, that is true. So Dr. Nasser is asking about Streptococcus sanguis. I'm not sure in what context.
But,
Oh, okay.
Um,
And
as a,
yes, you want to comment something?
No, I think some other doctor has asked, as a private practitioner, how do I apply this in practice? So see, if you look at takeaway points from this from a clinical practitioner, if you look at it, very rarely you will find people with periodontal disease also having a lot of dental caries. And when I'm talking about dental caries, it does not mean occlusal caries. So occlusal caries is directly as a result of food. But when you look at smooth surface caries and interdental caries, that definitely when you find that there is something wrong in the system. So that is something that needs to be investigated. When you also look at patients who uh, have this kind of periodontal disease, it might be especially aggressive forms of periodontal disease. As form of a therapy, maybe at some point we try to understand what are the etiological factors. Like you said, it's not just the bacteria. Today, bacteria account only for initiation of disease. Progression of disease is completely a host-dependent mechanism. So there are ways of coming up with host biomodulators that are available in the market to help your patients overcome the disease and also maintain them. So just like we have over-the-counter mouth rinses that just focus on antimicrobial effects, we could have some therapeutic strategies that help in controlling or biomodulation of the immune system that keeps the system at bay, the disease at bay, especially in patients who are susceptible to severe forms of periodontal disease. So I guess that is something that as practitioners we need to look at. So.
True. I think one good thing to do would be to study the periodontal microbes for their properties that could stop caries, and the reverse, and the cariogenic microbes for whatever properties they have that could be antibacterial towards the periodontal, because clearly they don't play well together. So maybe they have something that stops the other,
You know,
and maybe we can isolate and use that.
ma'am, I think you've hit the nail on the head here. And I'll just give you a brief insight on in our own lab. In our lab where we used to work, there was another a very respected professor, Ramsubbu Narayan. He is working on a compound. So what AA does is when it forms biofilms, and when you look at aging biofilms like 2 year or 2 day old, 3 day old biofilms, the A escapes from the biofilm. So when you look at it, when you look at the colony, it looks like a Halley's comet. So when he found this out, and this Dr. Ramsubbu along with Dr. Jeff Kaplan discovered that A actually produces a chemical, a protein that destroys biofilms, and they published that paper, and it's called dispersin B. So they patented that, and they purified that protein, and it eradicates a significant amount of biofilms, not just oral. So it's now been patented, and actually one of the chemicals that's available, like pulmozyme, it's like this, that it helps in prevent of joint replacement infections by preventing the forming of biofilms on these artificial hip joints, on these cardiac stents and stuff. So coatings of these have been done. So definitely, like you said, use the bacteria against themselves. So learn from them, try to get some. And I think Prerna,
Yeah.
I don't know if she's listening to this, she also played a lot of effort and did a lot of work on this as well. So yeah.
Prayer. Yeah. Prayer. It might be night there for them, right?
No,
She's
Prerna
asleep.
messaged me that she'll be here. Prerna is one of the
Okay.
because she also worked on this.
Yeah.
She also did a lot of work. Yeah.
Yeah. I remember that this person B work was excellent. Yeah. Great work by Subbu.
Yeah.
So we have this one more question. What are the saliva bacteria that play key role in infection of root canal treated tooth, and what are preventive measures? I don't know if this comes in your area of work, but
Well, not that I am not much of the oral bacteria, but there is some nasal bacteria that have been involved. So when you look at MRSA, for example, Staph aureus, and some of the other bacteria, Econella, I think these are actually their reservoir is the nasal cavity. So what happens is, since the nasal cavity and the oral cavity are quite close, there have been studies which show that the nasal bacteria are actually being found in the canals of infected root canals. And these are, and they also actually possess some of these root canal infections, reinfections of root canal. Certain types of bacteria contain this Van Hack's operon, which is an antimicrobial resistance gene for vancomycin, which is our last silver bullet against against MRSA. And now we have VRSA as well. So the ecological shift that can occur between two very closely related or closely situated niches, the oral and the nasal mucosa, could play a key role, and reinfections of root canal could also be attributed to some of this. So
Yeah. If I can add to this,
yes, please.
as I said again, these most oral infections are not like other medical infections with single species. There are so many. So much is known about the microorganisms, bacterial species involved in endodontal infections, but Enterococcus fecalis is one of the predominant ones.
Yes.
It is present in quite high numbers, and its role has been demonstrated in many in vitro and in vivo studies. It produces a lot of virulence factors. So if that could be targeted, like any antibiotics. So that is one thing.
Okay, so I don't think we have any more questions. We have, of course, a lot of thank yous and how great the presentation was. So this one is from Dr. Sunita, who says, "Thank you for an informative lecture." And earlier on, a lot of other colleagues have also said very nice lectures. And Dr. Nasser says, precisely to the discussion, it was all very correct. Let me see if I can pull up any more comments for you all. Yeah, that's about it. Okay. And earlier on, Dr. Meghanand also had said excellent presentation. And Dr. Nasser also. So most of it is just a lot of hellos to you. And okay, so we shall now, I think, give me a minute and I shall share your certificates and right. Yes. So thank you so much, Dr. Krishnaraj. I know today was rather inconvenient for you, finally, unexpectedly, but you sort of did a great job. You made it through. Thank you so much.
No, ma'am. Actually, I'm here. We came here to vote for the Senate elections are happening today in Rajiv Gandhi University, and the Senate members have promised that they are going to take up this issue of getting this dental insurance also from the university to
Oh,
the dental council. So yeah,
Yes.
that's one of the agendas. Yes.
Yes. That is very nice. Finally, getting people to promise things before electing them is always a good thing. Yes. All the best to you. Yes.
Thank you.
So, okay, the next one is, of course, for you. Thank you, Dr. Maribosappa. It was very kind of you to come and join us today.
oh,
And yes, of course,
yes,
now everyone knows that you're working in this field, so hopefully they will get in touch with you and you can help out most others who are interested.
yes, sure. It was my pleasure. I was so happy to do this for my
Thank
good
you.
friend Raj.
Thank
Thank you,
you
Raj.
so
Yes,
much.
yes, thank you.
Yes. So,
All right.
one
Thank you, ma'am.
second.
Thank you.
You're welcome. Just
Thank
two
you. Thank.
minutes more till I finish. Yes, yes, you were
Yeah,
saying, yeah, I know you wanted to say thank you.
I
Please take the minute. Yes.
yes. I also should thank you. I didn't know you, but I now came to know you through Raj, and I should thank you. Thanks for the opportunity to participate in this. And it was again like refreshing. I knew most of Raj's work, but yes, his thoughts and views of research, this research area, it's always like renewing. It renews my interest in the research area, this particular area. Thank you. Thanks,
Thank
Raj,
you.
and thanks, madam. Yes.
Thank you, ma'am.
You're
Thank you,
most
madam.
welcome, and it is definitely great that you could be with us. And of course, all the viewers who come back week after week, we have some regulars. We also have who, comment and then we know that they're there. But we also have many who don't comment and we don't know they are specifically present. I can only make out from the numbers, but every bit of support counts. And I have to say thank you to all of you. And now coming to some other sort of information, among those being that we have now the short videos that you can submit to the channel and it will be put up. It will not be live, it will be recorded. All of these series that I have listed there, you can submit your 15-minute videos. It will have to be submitted as a proposal first, basically just telling me what it is about, and preferably the details are given and the link is in the description. So essentially, you need to get me your presentation, all the text and all the PowerPoints from the word hello to the thank you at the end. We go through it, and if it is suitable, I will also have experts who will go through it. If it is suitable and it passes the initial review, then we will help you make the video. So don't worry about the video. Okay. And so that is one thing. And the other thing, of course, is our ongoing contest, which is still on for a few more days. So if you still want to submit your stories, please go ahead. It is stories not about the science of dentistry. It is about the human stories around that, your interactions, your personal stories that you built with experiences that you had, whether it was with colleagues, with students, with teachers, with friends, with patients. I've got some very interesting stories so far. So I hope you will also submit yours in quite soon. And of course, there are many prizes, and the link for this also is in the description. Then coming to next week's program, next week we have a legend with us, actually a special guest. It is the series is oral pathologists in focus. Every oral pathologist and probably many dentists also who have gone through his hands, because he was the dean for a long time, Everyone knows, of course, we all know Dr. G.S. Kumar. So it is it is great. We are going to talk to him. He is going to discuss his experiences as an oral pathologist, as a teacher, and what are his visions, what are his thoughts, and all of that is going to be there with us. And I hope you will join us. And I also hope that if you like this, you will hit like, and also you will share it. You will subscribe to the channel if it's your first time, and also comment. Your comments, if you let me know what sort of things you like, what sort of things you don't like, maybe I can modify all of those. So, with that, and I am going to say bye and have a wonderful week and see you coming Tuesday.